In this video, we're going to take a look at some of the different treatments of peptic ulcers and how some of these medications work to reduce the acid or release the acid into the stomach. So, important to note here, stomach acid is produced by cells that are called parietal cells, and these are found in the lining of the stomach. And so the peptic ulcer treatment does involve regulating acid levels in the stomach. And there are two kind of main approaches. One is to stop the production of the acid altogether. And the second is to prevent the release of the acid into the stomach. So let's take a look at some of the medications that are used to do these two different sorts of approaches. The first one we're going to look at is ranitidine. I think that's how it's said. Otherwise, it's known as Zantac, and so that's kind of a little bit easier to say. And so this particular medication inhibits the production of acid, and it does that by binding to receptor proteins. They're called the histamine H2 receptors in the membrane of parietal cells. And so what this does is it stops the normal chemical messenger, the histamine, from binding to turn on the chain of events that's going to eventually result in the production of acid. So otherwise, it's known as an H2 receptor antagonist, which means it blocks it from doing its job. The structure of Xanax is given here, and so it's worthwhile just stopping and taking a look and seeing what functional groups can you find, as well as are there any chiral centers in this molecule. So make sure you do that and take a close look at this molecule. The other classes of medications here, I'm not even going to attempt to say the names because I don't know how to pronounce these, but they're commonly known as Losec or Prilosec is one class, and the other class is Nexium. So these are specifically proton pump inhibitors, and they prevent the release of acid from the parietal cells into the stomach. So this is the second method of treatment for peptic ulcers. They are weak bases, and what's really neat about them is in the blood, they are unionized, so they don't have an extra proton on them. That means that they are nonpolar or lipid soluble, so they can cross the cell membrane of the parietal cells. And then when they're inside, they'll become protonated, so they'll gain an H plus inside the parietal cell because the environment is so much more acidic. And that protonated form can then bind irreversibly, and that's really important, to the proton pumps. And it's effective until the cell produces new proton pumps, and the cell does continuously produce new proton pumps, so you do have to take this medication over time. The two sort of structures here. So the first one, this is Prilosec. Just important to keep in mind, Nexium is just a stereoisomer of this. And then this is the unprotonated form. So you can see on this particular structure that this nitrogen here is unprotonated, but then in the active form of Prilosec, the nitrogen here is now protonated, and it's joined to create this ring structure here. So there's some other sort of chemistry going on here to create that particular ring structure, but just kind of important to note that unprotonated, and then this one is protonated. This is essentially the diagram of a parietal cell. So just kind of keeping in mind, this is the inside of the stomach here. And so the gastric gland lumen, this goes out to the stomach. And this is where the acid is coming from and going into the stomach. Now, the H2 histamine receptor antagonist, so this is histamine here, and it's going to bind to the histamine H2 receptor. But our H2 receptor antagonist is going to block this process. So they're going to come in, they're going to bind to this receptor here, which essentially blocks this pathway. So that means all of this then downstream is blocked, and the proton pump here is blocked. The proton pump inhibitors, because they are lipid soluble, they're able to cross the cell membrane. And then once they get in here, they become their protonated form. And their protonated form then is going to come and bind to this proton pump, which is going to block it and block the release of the HCl into the stomach. So just a couple of different ways and a nice sort of visual of how this particular system works. Now, the proton pump inhibitors, those are considered to be active metabolites. And so these are basically different forms or active forms of drugs after they've been processed in the body. And we have seen a few different examples in some of the other videos that we've looked at. For example, codeine will get converted into morphine in the body, which is going to then bind more strongly to opioid receptors. So that's one that is an active metabolite. The two different proton pump inhibitors are going to get converted into protonated forms, which then are able to bind to the proton pumps. And then aspirin is also converted into its active form, salicylic acid. So I think it's kind of neat to see these different types of drugs in their active metabolite form. And some of the interesting reasons why we want to create these active metabolites. In the case of aspirin, it's to avoid really negative side effects. In the case of the proton pump inhibitors that we just saw, as well as diamorphine, it's so that the drug can pass through the cell membrane first and then get converted to its active form. It can also, other reasons why you might want to have a different form to its active metabolite is to allow the drug to dissolve in water more easily, as well as to target drugs to a particular area. So things like the proton pump inhibitors, those do require acidic conditions to be activated, and that's only going to happen in the parietal cell. So everywhere else in the body, there's going to be essentially no effect. So those are some different ways that we can block the production of excess acid in the stomach, as well as what active metabolites are and how they work in the body. That's it for this video. We'll see you in the next one.